Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jonas Buge from Thermo Fisher Scientific.
Thank you very much for the introduction, Leah. Hello, everyone, and welcome to today's webinar. Today, we are going to discuss a number of topics that I hope will help simplify or streamline the analytics involved in testing for residual DNA impurities in a typical gene therapy process. Prior to that, I'd like to introduce you to our capabilities. Here at Thermo Fisher Scientific, we value our ability to support your gene therapy programs. That support goes from plasmid development through viral vector production, subsequent purification, and analytics. We have a broad portfolio of products, services, and a team of cross-functional bioprocess experts to provide solutions throughout your journey. We as a pharma analytics team focus on supporting the process analytics and product quality testing. Let's take a look at the specific analytical technologies used by our group in designing these analytical workflows. In this webinar, we'll focus on the current state of analytical methods in the gene therapy workflows and how leveraging the expertise and solutions Thermo Fisher has already developed can help simplify the burden on analytical development teams. All of our solutions facilitate one of two highly advanced and well-established core technologies, DNA sequencing and real-time PCR. Our MicroSeq microbial identification system utilizes gold standard genotypic DNA sequencing technology, providing accurate identification of fungal and bacterial contaminants as part of an environmental monitoring plan or a robust microbial risk mitigation strategy. Our solutions for detection of adventitious agents such as mycoplasma and viruses and quantification of residual host cell DNA utilizes real-time PCR to achieve fast, accurate, and reliable results. In the next slide, we will go over some current trends in the gene therapy field. Gene therapy is advancing rapidly, and we see remarkable viral vector-based drug approvals. Two notable AAV-based therapeutics are Luxturna for treating Leber congenital amaurosis type 2 and Zolgensma treating spinal muscular atrophy. There's an increasing number of clinical trials in the pipeline reflected in the 900-something applications the FDA currently has to investigate. There's also a corresponding increase in investments in this area. The market is estimated to reach $3.24 billion in 2024, just in 2 years, and double again by 2023. Some of these therapies are showing tremendous success at curing genetically related diseases. However, the risk of adverse effects in clinical trials has alleviated the urgency in addressing the safety and efficacy of these viral vector-based gene therapies. Some can be likely addressed by analytical teams in defining and monitoring the CQAs of the drug product itself. This will require employing advanced and perhaps novel analytical techniques by analytical development teams, especially in comparison to the analytics applied to the characterization of traditional biologics such as monoclonal antibodies. Recently discussed during a 2021 US FDA meeting were two topics that were calls to action for analytical teams developing viral vector-based gene therapies. The first one is about potential vector-mediated integration of non-vector DNA and the risk of oncogenesis. Assessing this risk requires analysis and elimination of sequences and vectors that increase the risk of oncogenicity. The second one is about whether an upper limit should be set for the goal vector genome dose per subject, and should there be an upper limit on the total capsid dose, given the presence of empty capsid impurities. In order to answer this, we need a more comprehensive characterization of empty capsids and other byproducts of AAV manufacturing. Designing good analytical methods to answer these questions remain a challenge in the industry. In today's webinar, we will only be addressing the first topic about the capability of our products to help you better characterize nucleic acid impurities and save you the challenge of developing these analytical solutions in-house. In general, what are the options when addressing the demands of analytics for gene therapy? First, you could develop an in-house method, which would require specialized technical expertise and extensive time and financial resources to develop, qualify, and validate the assay and to maintain a validated stage. You could contract a service lab to do the testing for you. This usually comes at a higher cost per result and adds to the timelines from sample taking to results. However, if you're just getting started with your project, contract labs might be a reasonable choice. Many contract labs use our solution, so you could easily start with a contract lab and bring the testing in-house later without changing the method. Or you could just start with our applied biosystems analytical solutions that provide fully integrated platforms for in-house testing. The general trend in the industry is to use a suitable commercial solution as early as and wherever possible in order to save time and resources to focus on analytical challenges specific to the process. We have also seen a large number of customers from all sizes to switch from existing in-house developed solutions to our commercial kits to utilize our validated workflow and the ease of use of our kits. In addition to the areas of increased concern and scrutiny, there are already a myriad of analytical assays for characterization and release testing for viral vector products that fall on analytical team's shoulders. Shown here is a list of expected tests for viral vectors. Most release assays can be categorized as either strengths, safety, or purity assays. Some of these are specific to each process or drug product and therefore need to be developed in-house, while others, such as mycoplasma detection assays, are more universal and not specific to a particular process or product. As such, internal resources of viral vector therapeutic manufacturers should be safe to focus on the specific needs of each product and process, and the evolving analytical needs to address the safety concerns. Our analytical assays, and even better, entire systems from sample preparation to instrumentation and 21 CFR Part 11 compliant software have been designed and optimized for the cGMP environment to meet regulatory requirements. Here is another view of the analytical assays Thermo Fisher has developed to simplify in-house analytical development for viral vector manufacturers and that are designed to meet cGMP requirements. You are looking at a typical viral vector production process and where in the process analytical testing requirements are performed. The lot release test for mycoplasma is typically performed at the harvest stage or end of viral vector production. Given the short turnaround time, MycoC gives the ability to perform in-process tests as intermittent test points. This allows you to design a very thorough risk mitigation strategy to detect the presence of mycoplasma early on in the process and prevent further contamination of your equipment, facility, or personnel. The residual DNA quantitation is performed during downstream process all the way through fill and finish. And lastly, microbial identification is performed throughout the workflow using site-specific guidance for environmental monitoring. As I mentioned earlier, today I will focus specifically on the analytical needs of viral vector manufacturers around residual DNA process impurity analysis. I will present the performance of our solutions to simplify your development needs and to give you the in-house control to create rapid and actionable data. So why should you be concerned with the amount of residual DNA left in your product? We know that residual DNA can impact product quality, efficacy, and safety. Regulators around the globe require you to limit the amount of residual DNA in the final dose. The World Health Organization, for instance, indicates that the amount of residual DNA per dose is kept below 10 nanograms. Residual plasmid is part of the 10 nanograms per dose limit, along with host cell residual DNA. They also indicate a size limit of 200 base pair for residual DNA. Therefore, expecting a robust and validated method for residual DNA fragment sizing analysis. The FDA echoes the same guidelines and also emphasizes that host cell DNA content is kept as low as possible and that manufacturers are using a highly sensitive method to determine DNA levels. In traditional biologics production, meeting regulatory guidance on limits has not been a significant problem. DNA clearance is well established and characterized. But in viral vector production, this has become of increasing concern, primarily due to the potential encapsulation of the residual DNA in the final product. Oncogenic sequences are of specific concern, and various agencies provide regulatory guidance around it. The European Medicines Agency's guidance on the quality of gene therapy medicinal products, section 4.2.3, characterization of the drug substance, specifically indicates that the absence of known oncogenes has to be demonstrated, especially with viral vector products manufactured in HEK293 and HEK293 T cells, with the E1A and SV40 gene being part of the host cell genome, and the possibility of residual impurities being encapsulated. Key takeaways from this slide are robust analytical methods are needed to demonstrate that residual DNA is less than 10 nanograms per dose, the DNA size should be lower than 200 base pair, and there is no inclusion of known oncogenes in the final product. Rather than deal with all the time and effort developing, qualifying, and maintaining these analytical testing requirements in-house, we have already done one big part of the hard work for you. And we also support you throughout the process of validation and regulatory approvals. Our end-to-end solution consists of all-inclusive kits with a well-characterized standard and all reagents. It leverages an optimized sample prep system. We will talk about that later in this presentation. And all assays have been designed to meet the regulatory guidance with ultra-high sensitivity, reproducibility, and consistency. We are the largest provider of commercial solutions for residual DNA testing for biologics manufacturer and have multiple kits to support production systems with various host cell lines, such as CHO and NS0 common to mAb production, E. coli for the production of therapeutics and also plasmids, Vero for vaccine manufacturing, and a few more. I'd like to focus on the highlighted ones that we developed in recent years to support the gene therapy space and viral vector production. These are designed to meet the regulatory requirements in the HEK293 production system with our ResNAC quantitative HEK293 kit, our kanamycin resistance gene kit for plasmids, and the E1A DNA fragment length kit. For insect cell production systems, we developed a kit for the simultaneous quantification of SF9 host cell and baculovirus vector DNA. Shown here are those DNA quantitation kits specific to viral vector production. The key features of the assays are an ultra-high sensitivity with an LOQ of down to 0.3 picogram per reaction for the HEK293 and SF9 and baculovirus DNA, and 30 copies LOQ for our kanamycin plasmid resistance gene. A well-characterized standard to support consistent performance, a single source for all assay components with predictable quality from kit to kit, lot to lot, and year to year, and a rapid and streamlined workflow with optional sample prep automation and time to results of less than 5 hours. The streamlined workflow begins with a sample prep, where you can choose between manual and automated options. The option shown here is a semi-automated version, utilizing the Kingfisher flags for up to 96 extractions at a time, meaning that you could easily go through 500 samples per week. Regardless of the level of automation chosen, the sample prep always features a well-established Prep-seq chemistry, which is based on magnetical particle separation. The second and third step in the workflow is setting up and running the ResNAC assay on one of our recommended Applied Biosystems real-time qPCR instruments, and the results are reported in the AccuSeq software. A few words on AccuSeq. The AccuSeq real-time PCR software integrates with our recommended instruments and provides many features to support you with the setup, running and analysis of your real-time PCR experiments. The software has SAE functionality that helps enable 21 CFR Part 11 compliance and full traceability of all actions within the software. In the next few slides, we will discuss the need for a good sample preparation method and how our PrepSeq kit fulfills this need. The gene therapy sample matrices are typically associated with PCR inhibiting or interfering components such as benzonase, detergents, media components, and potentially high levels of other non-target nucleic acid material. We have tested our PrepSeq chemistry on these challenging sample matrices to ensure it functions with the high efficiency we expect. Here are various sample matrices from different stages of the production process, and results were within specification, even in the presence of intentionally added inhibitors. So how does PrepSeq work? PrepSeq involves a combination of alcohol precipitation and magnetic bead-based separation of nucleic acids from various sample types. This is a universal sample prep for extracting double-stranded DNA, single-stranded DNA, and RNA. It is currently being used for a variety of our applications. The PrepSeq kit has shown superior performance in obtaining quantitative recovery, high precision, consistent performance with complex matrices including low pH, high salt, and high protein. The vast majority of our assay customers are also leveraging this validated sample prep method. The PrepSeq chemistry can be used in both manual and automated workflows. Depending on the type of automation and experimental design you choose, you can process between 1 and 96 samples in one run. The manual method is low throughput and allows 1 to 16 extractions per day. The Kingfisher Flex system is high throughput and allows for up to 192 extractions per day. Besides the high throughput, the Kingfisher Flex offers many other advantages, such as easy setup using graphical interface, high-speed purification of nucleic acids. It controls cross-contamination thanks to the bead transfer technology. And lastly, you can expect a consistent, high-quality nucleic acid recovery from a wide variety of sample types. Now let's get into the specific assays we offer that apply to the HEK293 production system. Those include solutions to quantify residual host cell DNA and residual plasmid DNA, as well as our recently released E1A fragment sizing assay. This is a general overview slide showing the utility of the analytical tools we will focus on today. The E1A fragment sizing assay is suggested to be used to assess the efficiency of your DNA size reduction step, such as benzonase treatment, and at the final stages of the downstream processing. The HEK quantitation assay and the CAN-R residual plasmid assay are typically used throughout the downstream process to support the characterization and optimization of your process and for routine QC. Let's start with the E1A assay. It's our newest assay we've developed specifically for HEK293 processes to address the aspects of sizing and oncogenicity of the regulatory guidance that we covered earlier. This kit can simultaneously detect and quantify E1A DNA of 3 different fragment sizes. But what is E1A? E1A, or adenovirus early region 1A, is a gene that is present in HEK293 cell lines. This adenoviral gene fragment has been intentionally integrated into chromosome 19 and provides essential genetic regulatory modulations for viral vector manufacture. It's because of this gene that HEK293 and various related cell lines are used to produce recombinant adenovirus, recombinant adeno-associated virus or AAV, and recombinant lentivirus. A current challenge in viral vector manufacturing, and we discussed that earlier, is the co-packaging of host cell DNA within the recombinant viral vector capsid. Problem. As E1A is part of the HEK293 host cell genome and also a known oncogene, the potential residual E1A deserves specific attention of detection and quantitation as potential harmful process-related impurities. Regulatory guidance also requires a method to demonstrate the effectiveness of the DNA reduction process to fragments lower than 200 base pairs. Therefore, we develop a qPCR-based assay to simultaneously address both concerns by quantifying different fragment lengths of residual E1A. The design involves 3 singleplex assays targeting known overlapping fragments sizes of short, medium, and long fragments, or 86, 200, and 476 base pairs respectively. The assay requires 3 standard curves, one for each fragment size, to quantitate E1A fragments in unknown samples. Given the overlapping nature of the target fragments, the short assay quantitates all fragments larger than 86 base pairs. The medium assay quantitates all fragments larger than 200 base pairs, and the long assay quantitates all fragments larger than 476 base pairs. We will provide you with a calculation tool that helps you calculate the E1A fragment size profile in your samples. Similar to all of our assays, this kit comes with all reagents and standards. This being a kit with 3 singleplex assays, all 3 assays show high linearity and efficiency to enable quantitative results across a broad range of DNA concentrations. We have validated the kit in matrices used in gene therapy to reflect a typical application situation, including inhibitors and at various concentrations. The kit has shown excellent performance under these conditions. This slide supports the earlier claim of excellent standard curve performance. All 3 standard curves show good PCR efficiency of 90 to 110% and an R-square of greater than 0.99. These results meet the criteria described in USP 509 residual DNA testing chapter. Let's move on to our HEK293 assay. This kit amplifies a highly repetitive element in the HEK293 genome. It was launched in late 2020, and we have seen tremendous success in the gene therapy field, but also for vaccine and cancer drug manufacturing. The ResDNA-seq kanamycin resistance gene kit was launched in 2021 and has been implemented at a number of our gene therapy customers. Both these kits follow the same general workflow, which begins with a DNA extraction procedure using PrEP-seq, followed by a qPCR assay for quantitation of the target DNA fragments. The graph on the left shows the standard curve generated using serial dilutions of plasmid DNA, standard provided in the kit. The dynamic range reaches from 30 to 300,000 copies. The second one shows the standard curve plot of an HEK293 DNA standard, also provided in the kit, ranging from 300 femtograms to 3 nanograms. This data proves the high sensitivity and broad dynamic range of these 2 assays. The broad dynamic range allows testing of a wide range of samples. You can see that the method linearity within the dynamic range easily meets the specifications as demonstrated by the R-square value of above 0.99. In addition, the PCR efficiency has been calculated to 105.3% and 101.6%, which sits well within the target range. Let's take a look at some other kit specifications on the next slide. Beyond the specifications from the previous slide, we see that all assays have an acceptable precision, a limit of detection of 15 copies for the CAN-R kit and 30 femtograms for the HEK293 DNA kit, a limit of quantitation of 30 copies for the CAN-R kit and 300 femtograms for the HEK293 DNA kit. The assays have been validated using both of our recommended instruments, the Applied Biosystem 7500 Fast and the QuantStudio 5. All kits go through a very streamlined and rigorous internal validation process, in which we test some of the parameters outlined in the ICH Q2R1 guideline document. The validation design that we use is slightly different from what an end user does for validation. We evaluate precision, accuracy of spike recoveries, linearity, range, LOQ, and LOD. We also perform a very extensive specificity test using a wide exclusion panel. Just as an example, for our validation process for the E1A kit, we used 5 operators across 2 sites, 3 single-plex assays, 2 sample prep methods, manual and semi-automated, 2 instruments, and we performed the study in 4 days. These reports are available to anyone interested in evaluating our kits. You can reach out to your local Thermo Fisher representative or to our technical support team to view a copy of these documents. Our support goes beyond just providing a verified and validated commercial solution to address these analytical needs with confidence. Through our implementation program, our field application scientists are able to provide you guidance on method optimization, qualifications, and validations to accelerate the implementation and integration of these methods into your process and make them suitable to address the specific regulatory needs. Through our certified field service engineers and compliance specialists, we also offer IQ/OQ and computer system validation services to create a one-stop shop for all your needs. We have a growing team of global regulatory specialists that can assist you with attendance at Type C meetings, providing detailed responses to regulatory queries during reviews. For relevant products, we have a drug master file in place to allow for access by the regulators should they need additional proprietary information about our assays. We also offer special regulatory support packages to assist with any additional needs you may have. The regulatory team members are also actively engaged with industry organizations, Bioforum Operating Group, PTA, FDA Advanced Trial Testing Interest Group, MIT CAACB, and Biomed FDA NAST Standards Working Groups, just to name a few. Bottom line, you are in good and capable hands. Getting to the end of our webinar, I'd like to summarize our residual DNA quantitation solutions in AAV production by highlighting once again the key features of the system. The ResDNA system is an all-inclusive system coming with highly characterized DNA standards and all reagents necessary for residual DNA quantitation and analysis. It provides an optimized sample preparation for quantitative DNA recovery. It provides ultra-high sensitivity and specificity with no cross-reactivity to unrelated DNA. The streamlined workflow provides reliable data within 5 hours. Our quality assurance team is making sure you will be receiving the same high-quality performance from kit to kit, lot to lot, and year to year. And finally, our worldwide technical support network will support you throughout all phases of the implementation process, from early evaluation over qualification and validation all the way to routine testing. Thank you for taking the time to join today's webinar. We hope that this webinar was helpful in understanding how we at Thermo Fisher can help you simplify the development of analytical solutions in gene therapy and other modalities. With that, I'd like to turn it over back to Leah for any questions.
Thanks, Jonas. So we do have a few questions, and just as a reminder, you can go ahead and type your questions into the question and answer window. And if we don't get to your questions, we will be passing any questions on to Jonas directly. So the first question that we have is, can I use the fragment length assay targeting E1A DNA to quantitate total HEK DNA as well?
Well, the quantitative HEK kit is specifically designed to yield super sensitive quantitation of residual HEK 293 DNA, and it does that very good. The E1A DNA fragment length kit will provide additional information about the fragment sizes in the sample. The assay design has significant differences as well. The HEK 293 kit targets a highly repetitive conserved region of the genome in order to have the confidence of the sensitivity requirements to satisfy regulatory guidance. E1A is only present once in a gene. We do, however, have preliminary studies that show that robust correlation between the E1A quantitation and total residual host cell DNA for HEK 293 cells. Well, it's not a one-to-one relation, especially across different concentrations, but when looking at downstream clearance studies, we do see a similar order of clearance. That said, we have gotten that question from a number of customers, and all have gotten that decided that not to replace the HEK kit because of the desired level of sensitivity and that they can only get with the HEK kit. Then they added the E1A assay to their testing plan.
Do you have any idea what the regulatory acceptance is for the HEK 293 residual DNA kit?
Yeah, I have looked into that now that we have the kit for 2 years on the market and hundreds of customers. It's still hard to determine how many of them went through regulatory filing, as customers barely reach out for help, as the kits are so easy to implement. But what I can say is that we haven't gotten a single feedback from a customer that ran into challenges with regulatory bodies, which is also the case with all of our kits, by the way. I can say that we have a large number of customers that are using our kit quite extensively and buying large quantities very frequently. And I'd say it's fair to assume that this is a strong indicator for successful regulatory filing.
Okay. And can I use the kits on digital PCR instrument, or do you offer any available solutions for digital PCR?
Yeah, that's a hot topic right now. I must say our kits are designed to work very well on qPCR. Customers told us positive experience with some assays like the CAN-R assay, the E. coli assay, our E1A assay on digital PCR instruments. However, other assays with highly repetitive regions like our HEK assay, they did not work quite well, which I guess is not that much of a surprise given the functionality and how digital PCR works.
Can you tell me a bit more about the CAN-R kit? How do I know if my plasmid would be compatible with your kit?
Yeah, that's a good question. What I can say is we developed the CAN-R kit to pick up the 3 most common alleles of the kanamycin. And we analyzed the typical plasmids that are out there and selected a gene that covers the largest possible amount. And as a result, it does work very well for approximately 95% of plasmids that contain the kanamycin resistance gene. That said, in your case, it's very likely, 95%, that your plasmid will work. But to be certain, please send me or your tech support an email and we can have our bioinformatics team check it for you. And when I remember correctly, I've only experienced it once that a match was not as good. Customers still purchased the kit and results were still sufficient or satisfactory.
Okay, it looks like we're almost out of time for questions, so we just have one more question for this webcast. Go ahead and continue to type your questions in, and we'll pass those on to Jonas. So the last question is, okay, is there cross-reactivity between this kit and the other non-HEK 293 cells?
Yeah, we've only mentioned that briefly earlier, right? But we do extensive exclusion panel testing to ensure our kits meet specificity requirements. That being said, our Restinase kits are species-specific, but not specific to a certain cell line. Therefore, the HEK kit will, as per the design, pick up and accurately quantify any human DNA and low level of target reactivity with Vero DNA, but that's not being a surprise. But for unrelated species, we have seen no cross-reactivity at all.
Thank you, Jonas, and thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. And just as you go today, you'll see a survey pop up. Please go ahead and answer those questions and give feedback to our presenters. Thanks again. Goodbye.